My name is Quentin Funderburg. I'm a rising junior at Williams College, and I joined the Gum Lab this summer to talk about Bryophyte Rubisco expression and assembly in E. coli. This right here is a structure of Rubisco. We'll be talking about the protein a lot, so we'll get into it a little bit later. But to start, just a quick overview. We're first going to look at the important context of Rubisco and why we want to study such a specific protein for plants, and then sort of how we can get into the ways in which we can study it so that we can flow into my project about Bryophyte Rubisco. So the enzyme Rubisco has a really long name. Rubisco is an acronym that means ribulose 1,5-bisphosphate carboxylase oxygenase. It's a mouthful, but that first word, ribulose 1,5-bisphosphate, is usually shortened to RUBP. It's a sugar in the Calvin cycle of photosynthesis and is the substrate of the enzyme. Carboxylase and oxygenase are its two functions, which it has due to its low specificity. The other substrate is a gas, either carbon dioxide or oxygen. And you can see in the carboxylase reaction, RUBP and CO2 interact through Rubisco to form two 3-phosphoglycerate molecules, which is our target product that we want. But in the oxygenase reaction, if O2 interacts, we have one less carbon, and so therefore we get one molecule of 3-phosphoglycerate, but then one molecule of 2-phosphoglycolate, which is toxic to the plant and must be metabolized in photorespiration, which consumes energy and emits previously fixed CO2, which is never good in terms of that's the fact, that's the job that Rubisco does itself. The other problem with Rubisco is that it also is just a really slow enzyme. The chemistry involved is quite complex, so it takes a while. And therefore, plants try to compensate by overexpressing that protein in order to meet the carbon load that they need to get from the atmosphere. And so you can see these plants here have some outrageously high amounts of Rubisco in their leaves. Up to nearly a third of all of the nitrogen in that leaf is going towards Rubisco. So an intense amount of energy for the plant is going towards one enzyme. And so as a sort of big picture idea, scientists want to find enhanced Rubiscos in species we haven't seen or engineer a Rubisco that can be improved so that it can be put into things like agricultural crops so that we can increase yields due to like metabolic efficiency changing with an enhanced Rubisco. So now that we've sort of gone over a big picture, we're going to sort of look into ways we can study it. And that really comes down to looking at the structure of proteins, because that's how proteins work. Their structure is really fundamental to their understanding, and biogenesis. So it is a hexadecameric complex, which means it has 16 components. There are 8 large subunits, which are around 50 kilodaltons, and they will associate into L2 dimers. And then we'll also have 8 small subunits around 15 kilodaltons, which will cap the final enzyme right here, which we call the L8S8 complex. It is a very big protein, that is all I can say. However, it can't actually assemble like that on its own. It has a ton of helpers. So you can see this really complex diagram is from a paper that developed a method to express it in E. coli, but we have the chaperonins, which help fold the large subunit. It needs its own environment to fold. And then the rest, these assembly proteins, help facilitate bringing the large subunits and the small subunits together into that final complex. These papers here, starting from 2017, developed a method to express in E. coli. Of course, that biogenesis process happens in plants, but to better study it, it's easier to do it in E. coli. And what they basically found most crucially is that you have to express all of these things. You can't just express those subunits. And so with that, you can get a ton of Rubisco, and that is what I'll be doing. So my project focuses on this expression system in order to optimize and sort of stretch the limits of what we can do with it. If I am specifically concentrating on Bryophyte Rubisco, and so a brief intro to what Bryophytes are, I'm sure Fei-Wei Li is excited to hear. They are a deeply diverged group of land plants. They diverged from the rest of land plants nearly 450 million years ago. They're a monophyletic group, meaning that the 3 groups, hornworts, liverworts, and mosses, are all related and have a common ancestor, which means it's helpful for my project and that they're closely related. Some interesting physiology is that they're non-vascular and seed-free, but generally they're quite understudied, and so there's plenty of things to be looking for, especially their Rubiscos. I'll be using the model species because they have annotated genomes online, which is quite easy for my job, and I'll be abbreviating them as such here. So when we're trying to express these things, the Rubisco in E. coli and the other proteins, we have this plasmid system developed by a previous researcher in the lab. So we have one plasmid for the large and small subunits, a plasmid for the chaperonins, and then a plasmid for all the assembly proteins. For the best results and best yields, you'd want to use sequences from the same species. However, for my project, that's what I'm trying to sort of stretch. So my control we'll be looking at trying to produce hornwort Rubisco using hornwort chaperonins and assembly proteins. And that is the system we currently have in our lab. But I'll be trying to make liverwort and moss Rubiscos using those hornwort chaperonins and assembly proteins just to see if it's possible, so we can facilitate making more Rubiscos of different types to study them just quicker and faster. So an overview of my methods. Once we have those plasmids, we can transform them into bacteria, grow them up through a series of cultures and antibiotic selection so that we get some E. coli with Rubisco in it. And then I can take that Rubisco, extract it out of the cells using a French press cell lysis, and then 2 stages of chromatography. I'll be using a nickel resin in order to capture them from the rest of the cell lysate. And then I'll be using size exclusion chromatography in order to separate that from the rest of the proteins. And I'll be using SDS page and native page in order to analyze my results in this presentation, which is why I want to give a little overview just for anyone that doesn't understand what it is. So SDS page and native page are both ways that we can sort a group of proteins from a sample through running it through a gel. SDS page uses a detergent which will denature the protein so that they sort specifically by their molecular weight, seen here. And then native page instead keeps that native form, and we will again sort by size. But then what is most interesting is that if the native complex are the same size and have a different charge, they will show up at slightly different places along the gel, which is important when comparing something like an Arabidopsis rubisco to a Rubisco from another species. So now in our small scale analysis, this is after the nickel resin. I didn't do size exclusion chromatography yet. We get this funky gel, lots going on, but to try and describe, each 4 columns are correlated to the 3 species I was looking at. We have S, UN, W, and E. That's soluble, unbound, wash, and elute. What's most important is the purification process I go through, and we'll be concentrating on the E column in order to understand if I did make Rubisco. So this is the SDS page, and so once you denature the proteins, you have the small and large subunits separate, and we can see up right around 50 kilodaltons, we have a large subunit band here, and then a small subunit band down here right around 15, and this is our hornwort control, nice and bright bands, we can really tell that it's there. If we're looking at liverwort, there seems to be a really thick band in the E column, but it's actually right around 60 kilodaltons, and that's the chaperonins. So not really much large subunit there. There is some, but it's really hard to see. And then down here, we do see some small subunit. Then if we look at the moss, we sort of get a mix between the two. We have a decently thick band for the chaperonins, but there is definitely a noticeable band for the large subunit. And then lower there, I promised you, there is some small subunit there. I think it leaked a little, but it is there. Since when we look at the native, we can see that the moss Rubisco did assemble. But first, looking at the hornwort in our E column again, we have this nice, thick, streaky band. That's the full complex, which is great. Our control worked, at least that worked. And then we can see and look in the liverwort, there's really almost nothing there. That thick band there, once again, is some chaperonins, but there's nothing there. And so we stopped looking at liverwort Rubisco from there. And then if we look at the moss, we have 2 bands here. And one of those bands, the fainter one, is the Rubisco, while the other one is most likely an alternate complex with some assembly protein. So from there, I decided to do size exclusion chromatography with the moss and the hornwort Rubiscos, and we'll analyze from there. When you do size exclusion chromatography, you get what's called a chromatogram. It splits your sample into fractions, which the fractions that had proteins were fractions 7 through 12. Oh, my bad. And you can see that there's 2 distinct humps. The hump on the left is what the chaperonins are, and then the Rubiscos are in the larger hump. That's just from previous experience. And we can see that actually correlates into the gel, where we have really thick bands that are the Rubisco complex in fractions 9 through 12. This is the hornwort. We have really thick L8, S8, really noticeable large subunit and small subunit bands. So all good there on the control. And if we compare it to the moss, this is the chromatogram overlaid. We have the hornwort in the blue and the moss in the orange. And we can see chaperonins are at similar levels, but Rubisco for hornwort is much higher than the Rubisco for the moss, which is sort of to be expected since I used hornwort assembly proteins. That makes it quite hard to make any Rubisco, but what's most important is that it's still there, which is great. So when we look at the gel now, you can see we have really bright bands up there that are the chaperonins, like I mentioned before, but there's still those faint bands that we saw in the previous gel for moss. And that one above is the moss Rubisco in its complex form. And then below, we can see the large subunit and the small subunit. So it definitely got made, which is fantastic. So as a summary, I attempted to express liverwort and moss Rubiscos utilizing those hornwort chaperonins and assembly proteins. And the liverwort didn't assemble, but the moss did, which is great. It's the first moss Rubisco to be assembled in E. coli, which is a big yay for me. And yeah, there were some low yield, but it still happened. Some future directions, they're quite numerous, but all I did was actually make the Rubisco and find it, but you can still do kinetics on it. So since bryophytes are quite understudied, it's really good to do kinetic assays on the Rubisco to see if it's actually a Rubisco with really good kinetics that we could put in plants or not. And then after that, you could also change the plasmid source. So I mentioned I was using hornwort chaperonins and assembly proteins, but I could also use the assembly proteins from moss or hornwort, since those seem to be the ones that had the least sequence similarity to the hornwort ones. And using ones from the same species could hopefully improve our Rubisco synthesis. And then lastly, the small subunit. Every species has plenty of copies of small subunits. I think my moss had like 20 and my liverwort had like 7. So you could also exchange those sequences and see if you could improve the amount of Rubisco you get from that. So yeah, I'd like to thank Laura Gunn and Fei-Wei Li. Laura Gunn is my main PI, but I work in Fei-Wei Li's lab. Zen is my mentor. Bryce is the other mentor that I talked to quite a bit. And Tori was the other RU student in my lab. And Jen Hong is the person I did. She did the research before me. So thank you, everyone. Questions?
Hi, Quentin.
Hi.
Wonderful talk.
Thank you.
I was just wondering if you could tell us a little bit more about the significance behind your study.
Yeah, so at least in the Laura Gunn lab, of course I was speeding through it in the presentation, but our main goal sort of outside is to get as many Rubiscos as all, sort of a gotta catch them all mindset. And so we want to basically, using this synthetic biosystem allows us to hopefully be able to sort of make any Rubisco possible. And because there's so many different things you need to express in order to make Rubisco, it'd be better if we could just use the same chaperonin system from one species to make tons of Rubiscos. And so it's sort of an optimization to see, can we make this more efficient? Yeah.
Thank you so much.
Of course.
Great presentation. Really loved hearing about it. I was just wondering if you knew of any previous studies that indicated that the Rubiscos found in hornworts, liverworts, and mosses was more efficient than the types found in like tracheophytes.
Yeah, that's a good point. So the reason we're studying bryophytes, first off, Fei-Wei Li has the most hornwort biodiversity in the world, so great right there. But on top of that, they have purinoids, which I think is something that Tori mentioned. They're a carbon concentration mechanism, which is sort of another way that plants have adapted to put more CO2 near Rubisco. And so some preliminary studies have shown that some bryophyte Rubiscos have good kinetics, but generally we just don't know the kinetics. And so it's sort of a let's look because no one else has ever done it. So yeah. Awesome. Thank you, guys. Was someone? Could I miss? So sorry, I didn't see you.
You're okay. You're okay. Great presentation.
Thank you.
I just have a quick question
Yeah.
about, you talked about the future directions of doing kinetic studies. I was
Yeah.
curious, what do those kinetic studies kind of look like? Like what kind of techniques would you be doing in the future to study
Yeah.
the kinetics of the Rubiscos?
Yeah, so I never had any experience doing it, but Zen, my mentor, was doing it. He uses a bunch of radioactive assays. Usually, I think, I won't go in further because I probably will say something wrong, but we use radioactive assays in order to track the amount of CO2 or amount of reaction occurring in solution. So yeah.
Thank you.
Uh-huh.